Thank you for the introduction. So I'm a third year PhD student working at the IRDL with Stéphane Bruzaud in Lorient, but also at the LOMIC in Bagnols-sur-Mer with Jean-François Ghiglione. And today I will talk to you about poly-hydroxyalkanoates, that I will call PHA because it's shorter, and production and biodegradation. So for the one who don't know what is a PHA, this is the small pellets we can find in some bacteria body, because when they will be in an environment rich in carbon, they will store it as a carbon reserve. So this is basically a survival mechanism for them, but for us, it is mainly a polymer. Here you can find the chemical composition of a monomer of PHA. So we can see ester function here, because it's a polyester. But the most interesting thing about PHA is this radical chain here, which can change in number of carbon in size. So we will divide PHA according to the number of carbon on this radical chain. If there is 1 or 2 carbons, we will call them short chain PHA, and we will find 2 types of subunits. HB, hydroxybutyrate for 1 carbon, or hydroxyvalerate HV when there are 2 carbons. The second type of PHA we can find is when they have more than 3 carbons. This is the medium chain PHA, and we can find many subunits in those PHA according to the number of carbons still. And we will find homopolymer of short chain, like PHB, 100%, or we can find different copolymer composed of HB and HV, or copolymer of medium chain composed of HO or HHX. And the thing is, according to the composition of this polymer, the type of the polymer, we will find different properties, physicochemical properties, and we can find various applications to the PHA. Usually we find the short chain PHA in packaging application, while we will find the medium chain PHA in some biomedical application. Additionally, to be bio-based and coming from bacteria, they are believed to be biodegradable. So here is a study that has analyzed the biodegradation of a copolymer of hydroxybutyrate and hydroxyvalerate. And you can see on this picture, after 9 months in seawater, the polymer disappeared. The fact is, about the biodegradation of PHA, main studies are conducted on short-chain PHA because they are commercially available. And secondly, there are few studies in natural marine environments. So in this study, we aim to compare biodegradation signals and bacterial composition of various PHA that displayed various characteristics and properties. To do so, we had to produce different PHA and a set of PHA with various properties. So we produce the PHA by bacterial fermentation in bioreactor in Lorient. For the short-chain PHA, we used Halomonas, a species that is coming from Iroissé in Britain, and different substrate, different carbon source, so they can accumulate the PHA. So from glucose, we produced a PHB. From a mix of glucose and valeric acid, we produced a PHBV6 composed of 6% of HV content. And from another mix of glucose and valeric acid, we obtained a PHA composed of 11% of HV content. For the medium chain PHA, we used another strain, which is very well known to produce medium chain, which is Pseudomonas putida, from fatty acid, because it's going under the fatty acid beta oxidation process. And from octanoic acid, we had a PHO, mainly composed of HO monomer. From nonanoic acid, we had a PHN, mainly composed of HN monomer. And we mixed nonanoic acid and acrylic acid to produce another PHN, but with higher content of HN monomer composition. I won't get into the details of the characteristic of these polymers, but mainly what we have to remind and to understand is that short chain are more crystalline polymer, semi-rigid, a bit friable and brittle. On the other hand, medium chain will be amorphous polymer, rubbery, hydrophobic, and a bit elastic. Then we analyzed their biodegradation through a biodegradation experiment. And what we wanted to be is to be as near as possible as the natural environment that we can find in the sea. So in Bagnoul-sur-Mer, we did the biofilm selection. We wanted bacteria very specialized in PHA biodegradation or polymer biodegradation. So we have these we have these aquariums in Bagnoul-sur-Mer that are directly connected to the sea. So we have pump coming from the sea and giving water to the aquarium daily with all the bacteria and the microorganisms. We put 4 polymers, PHBV for short chain, PHO for medium chain, cellulose as a positive control, and polyethylene as a negative control. And we put these polymers in the aquariums for 1 month to get a mature biofilm specialized to the polymer. After 1 month, we detached the bacteria, and we inoculate new tube with the polymer previously produced, according to the right inoculum for the polymer. So the tube contained disc of PHA in minimal medium, and PHA was a sole carbon source, so the signals we will get are the biodegradation of the polymer from the microbial community. This lasted for 3 months, and during sample type, we looked at the oxygen consumption and the bacterial community by sequencing the 16S rDNA. The parameters were the same as the Mediterranean Sea, with the temperature of 18 degrees and a little bit of agitation. So here is the microbial activities showing the oxygen consumption of the 6 PHA, but also the control, cellulose and polyethylene. What we found out at the beginning is that no differences were observed within a PHA type, I mean significantly. There is no difference here when we look at the short chain. This is PHB, PHBV6 and PHBV11. There is no differences, but they are all above the positive control, which is here, the cellulose. Then we found the medium chain here, PHN, PHNAC, and PHO, and at the bottom, this is polyethylene, which is non-biodegradable. So here we see a high oxygen consumption for cellulose and less oxygen consumption for medium chain. Then we looked at the bacterial community composition. We performed a simple analysis, which means here is the taxa that will explain 50% of the dissimilarities between 2 groups that we compared. So we compared the short chain and the medium chain. We compared the short chain and the polyethylene, and the medium chain and the polyethylene. Here, the circle represents the size of the relative abundance. I mean, the size of the circle represents the relative abundance, and the color represents the contribution percentage from each taxa. What we saw on short chain and compared to medium chain, but also compared to polyethylene, is that Marinobacter species are highly abundant on this polymer and highly contributive, about 20%. When we looked at the medium chain and the polyethylene, the structure is more scattered, but still, for the medium chain, compared to the short chain or compared to the polyethylene, one taxa have been highlighted. It is Pseudomonas species, which is quite interesting because we don't know a lot about medium chain biodegradation in sea environment, but we know that Pseudomonas is a good producer of PHA. To conclude, we achieved the production of different PHA from different strains and carbon sources with various properties. Secondly, short-chain PHA have far more bacterial activities than medium chain when we looked at the oxygen consumption and the non-biodegradable polymer polyethylene. And we found a more specialized microbial community on short-chain than other polymers, while the bacterial community structure of medium chain were closer to PE than short-chain or polyethylene. Which is quite surprising because for polymer experts, when we looked at the polymer biodegradation, the more amorphous is the polymer and the more it is believed to be biodegradable. And this is not the case for short chain. So we still have a lot to understand about the relationship between the pH properties, these amorphous and crystalline regions they can display, and the biodegradation, looking at the extrinsic properties, which mean properties relative to the environment. Maybe short-chain PHA and medium-chain PHA depolymerase occurrence in natural environment are not equal. Maybe there are more short-chain PHA depolymerase than other depolymerase. We also plan to look at functional genes involved in biodegradation. And about the polymer part, there is these interesting properties that play a major impact on biodegradation. And we aim to better understand PHA depolymerase mechanisms on physical-chemical properties, but in vivo to look at the roughness of the polymer, how it can impact, and the structure, how it can impact the biofilm formation, and therefore the biodegradation. Finally, I would like to thank the people I worked with and the organizers, but also you for your attention.